Yes, perfect. Okay. Hello, everyone, and welcome to today's CGIL seminar. Today, we're delighted to have Dr. Christina Rochus, also known as Chrissy, who is a postdoctoral researcher at CGIL under the supervision of Dr. Christine Bayes. Chrissy received her Bachelor's of Science in Animal Biology and Master's of Science in Integrative Biology from the University of Guelph, a double PhD in animal breeding and genetics from the Swedish University of Agricultural Sciences, Agro Paris Tech, and INRAE Toulouse, as well as postdoctoral training at Uppsala University, Cornell University, and Wageningen University. Over her research career, Chrissy has worked with several different species, including Atlantic salmon, sheep, chickens, herring, pigs, turkey, and dairy cattle, with a focus on using genomic data to study genetic diversity and adaptation in domestic and wild populations. And with that, I'll pass it over to you to begin, Chrissy. Thanks.
Thanks, Colin, and thanks everyone for joining us today. So the work that I'm going to be presenting today is part of the work that I did while I was at Wageningen University in the Netherlands. Let's see. Yes. So I'd just like to start off by acknowledging my supervisor there, Han Mulder, and some of my other co-authors on a paper, well, a manuscript that's currently in preparation. This project was part of a Muta-Breed project, which was part of the Breed for Food Consortium, and two of the members of this consortium were Hendrix Genetics and Topics Norsvin. And so we actually, the data that I'm presenting today is data that we received from Hendrix Genetics and Topics Norsvin. But before we start talking about mutation rate and de novo mutations, I wanted to talk a bit about genetic diversity and where this interest came from. So during my PhD, I started getting interested in genetic diversity, and this was when I was studying domestic sheep, local breeds of Swedish and French breeds. And this allowed me to study signatures of selection and population history, which was really interesting for me. And then moving on to postdoc, I started to use sequence data to look at domestication QTL in chickens, and also in herring, looking at signatures of selection and population history. Now, this work is only possible because we really rely on looking at sequencing and genotyping variants. And these variants are really old variants that we're looking at when we're studying signatures of selection and population history. For example, in sheep, we were using a SNP array, and so this SNP array works for multiple breeds, which means that some of these SNPs are shared between breeds and must be thousands of years old. And it's the same when we were looking at the sequencing data, that when you're looking at domestication and you're also looking at some of these introgression events that we looked at between species, that these markers that we're really focusing on are really, really old. But what I want to talk to you today is more about where are these variants coming from, and how do they occur, and when do they arise? So this brings us to de novo mutations. And these are new mutations that occur in the germline, and they're occurring during gametogenesis, so during the production of egg or sperm cells, or it can also occur early in the fertilized egg cell, but it has to be really, really early in the fertilized egg cell so that it can be included in the germline and ultimately possibly passed on to the next generation. So if we were to look for these de novo mutations, what we would do is we would sequence a trio like we have here, and we would sequence just the somatic tissue. And in the sire and the dam, we wouldn't find this de novo mutation. And in the proband, we would. And this de novo mutation would be occurring in the germline, as I said, so that we could also possibly find the de novo mutation in the offspring of the proband, depending on whether they've inherited it or not. So de novo mutations provide new genetic variation, and like any other variant, they can have a range of effects. So it could be positive to neutral to negative. A variant could have a positive effect on one trait and negative effect on the other. And of course, what we really want to avoid are these negative effects that could really disrupt the biology of an individual and cause serious disease. But probably most de novo mutations are neutral or have really small positive or negative effects, and probably more negative effects because it's easier to disrupt this biology. But we want to keep these positive or neutral de novo mutations around because they help with adaptation or selection on future traits that we don't know about yet that could be really interesting, and then these de novo mutations could have an important effect on them. So in humans, de novo mutations are the best studied. So all of us, you and I, we have about 40 to 60 de novo mutations that are not from our parents. So if you were to sequence your parents, you would not see these de novo mutations. De novo mutations and the number of de novo mutations have been associated with several disorders in humans. So schizophrenia, heart disease, male infertility, autism spectrum disorder. And one of the challenges in humans is so far, they've really only been able to look for this association with binary traits. So they have thousands of trios which they sequence, but essentially all they can do is the parents don't have the mutation and don't have the disorder, and then the offspring has the disorder and has the de novo mutations. So this doesn't make for a very powerful study. But in livestock, this could be really interesting for us looking at de novo mutations and their effects on traits, especially if we might see something like an impact on male fertility as we've seen in humans. So unfortunately, in livestock, we've only seen one estimate so far of mutation rate, and that was in dairy cattle. This paper hasn't been peer-reviewed yet, but they found 35 de novo mutations per generation. So it's a similar mutation rate as humans, a little fewer de novo mutations per generation because the genome size is a little smaller. But livestock population structure is really helpful for estimating de novo mutation effects. So if you think back to the trio of humans I showed, when we start to think about trios in pigs, for example, so you have the parents who don't have the de novo mutation, you have the proband that does have the de novo mutation, and you can quickly breed that animal and produce a large number of offspring that are also carrying your de novo mutation of interest. And so in pigs, for example, you have a generation interval of about 2 years. So you can really quickly build up a number of animals who have this new mutation of interest, and also because there's a lot of phenotyping opportunities as well, measuring complex traits like production, growth, but also reproduction and functional traits as well. So I really think that this is an area where animal geneticists will be really able to add not only to our own field, but also to other fields like in humans. So we talked a bit about the potential effect of de novo mutations that could be positive or negative, but what happens to these de novo mutations in our selection programs? And this table here is results from a simulation study from Han Mulder, and he showed that simulating a number of different selection programs, mass selection and BLUP and GBLUP, he could show that the mutational response was actually the lowest in GBLUP with no own performance. And this really makes sense because de novo mutations are not in linkage disequilibrium with any SNPs that are used on SNP arrays for genomic selection. And if you're not including information on own performance traits, there really is absolutely no selection pressure being put on these de novo mutations. And in fact, those de novo mutations that do stick around in the population are just there by chance and by drift. So that raises the question, what are we missing out on? Is this a problem for future selection? That are we losing out on variation that could help us? So, but we needed to get to the basics here and to understand what are these de novo mutations? Can we find them? Can we estimate the mutation rate? And can we characterize these de novo mutations and start to get a picture of what's happening? So our study design, we had 41 trios from 2 commercial pig lines. And so about half were coming from Topix Norshin and half coming from Hendrix Genetics. The proband had over 30 offspring that had been genotyped with a 50K SNP array and also phenotyped. And this is for future work when we hope to estimate the effects of these de novo mutations we found. But it also puts a limitation on our study as well, because if a proband has over 30 offspring that have been both genotyped and phenotyped, there is probably no large effect, negative effect of de novo mutations on that individual. So we're not expecting to find any big negative effects here. So we used short read sequencing, and we had a depth of about 30x coverage. For our study design, we just used a typical single nucleotide variant and indel calling method from GATK. We kept only those single nucleotide variants, and then from those, we looked for de novo mutation candidates. And so the de novo mutations could not be found in any unrelated trios because the chances of one mutation arising simultaneously in the same generation in 2 individuals is probably quite low. Our read depth had to be between 10 and 100, just so that we had a lot of evidence for the de novo mutation that we found. The genotype quality, again, needed to be equal or over to 99%, just to make sure what we're finding is real. And we also looked at the quality in the surrounding 50 base pairs of each de novo mutation, and we used the number of variants within this window as a proxy for quality. So if a window had like 10 other variants within the same 50 base pair region, this region had really poor quality of sequencing. So other things that we needed to see, the sire and the dam needed to be homozygous for the reference allele. They couldn't have any supporting reads supporting the de novo mutation. The proband, on the other hand, should be heterozygous, and more than 20% of those reads should have been supporting the de novo mutation. Finally, we visually inspected variants using a program called JBrowse, and this is a step that most de novo mutation studies actually take, and it's because for some reason we're just not able to completely filter out all of these poor candidates just by using these different filterings. So here's a screenshot of what we see in JBrowse. So at the top here, you can see this is the reference genome. And then here we have our proband, our sire, and dam, and these are the reads. So in pink are the forward reads, in blue are the reverse reads. And then any place in the read that differs from the reference genome, you see it highlighted with a color. So you can see these different places are sprinkled throughout the reads of the dam, for example. These are all probably just noise, and these are actually the raw reads that we have, that I have here on the slide. So actually a lot of these reads probably would have been removed during our filtering. So here you can see our de novo mutation. So about half of the reads in the proband are supporting this T allele, which is different from the reference allele. And then you can see in the sire, it's homozygous for the reference allele. And then in the dam, you can see there's one read supporting this de novo mutation, but actually this read was removed in filtering because you can see how many different variants were occurring just on this one read alone. So this is what our de novo mutation looks like. So in total, we found about 19 de novo mutations per generation in each pig. Our mutation rate was 8.2 times 10 to the minus 9. And I've shown almost a complete list of all the mammals that have a mutation rate estimate. We're just missing a couple of different species of monkeys. So you can see that the pig rate is a bit lower than some of these other species. And I think that's because we were being very critical with our filtering steps, and yeah, our mutation rate is probably a bit lower than it actually is. One other thing that's interesting to note about these estimates, direct estimates of mutation rate, is that in humans, we have many studies that have thousands of trios sequenced. And so I think we can be pretty confident about this number here, 1.2 times 10 to the minus 8. In cattle, the number of trios was about 132. But in every other species, it's 8 or below. So really small sample size. So it can be difficult to make comparisons between species. But it's nice that we're adding with this study of 41 trios. There was variation between individuals. So we had a range of 2.6 times 10 to the minus 9 to 1.3 times 10 to the minus 8. And this again fits well into these other ranges that we see here in other species. I wanted to break down the mutation rate for you by line. So we found about half of the de novo mutations in line 1 and half in line 2 for a total of 704 de novo mutations. You can see in our line 2 here that we have a little bit lower mutation rate, and this is actually due to probably sequence quality. You can tell this from the transition transversion ratio, which should be about 2, and instead in line 1 it's 1.5. We think this has something to do with the storage of the samples that we used. They were semen. There were some semen used, and it could have been how we extracted DNA or whatever. But hopefully we fixed that problem now. Just to remind you about transitions and transversions, because it's really important for understanding the mutational spectrum. So transitions are these changes in nucleotides that have similar structures. So you can see this 2-ring structure of adenine changing to a 2-ring structure of guanine, or you have the 1-ring structure of cytosine changing to 1-ring structure of thymine, or could be vice versa. Whereas the transversions, there are many more possible changes, but it's changing from one structure, so it could be the 2-ring structure to 1-ring structure or vice versa. And so actually these changes take much more energy to change the ring structure. So we actually expect to see more transitions than transversions. So that's where this 2-to-1 ratio is coming from. So as I mentioned before, we do see this 2-to-1 ratio in our, well, close to the 2-to-1 ratio in our findings here of de novo mutations. But what's interesting is that when you start to break it down by the different base pair changes, that they're not all created equally. So you see that 50% of our de novo mutations, total de novo mutations, are coming from this C to T, whereas it doesn't seem like too many of our changes, our transitions, are coming from the T to C. And it's interesting to see this because this is different in humans, because there is a much higher rate of T to C. And this is actually probably due to the age of the parents. So in humans, they've found that age of either the father or the mother, as the age increases, the proband can have more de novo mutations. And they found that actually a lot of those de novo mutations that are coming from age-related reasons are these T to C. So we would expect our mutational spectrum to reflect more of a younger human, and this is what we see because in pigs, the age is about 2 years when they're bred. I've broken down these changes even further into, so you can see here our C to T, and then you can see I have shown the percentage of the de novo mutations that we found that were C to T that were surrounded by an A and a G. So 7% of the total number of de novo mutations were the C to T surrounded by an A and a G. So you can see that context also matters about where are these de novo mutations occurring and which de novo mutations are occurring. And I think that potentially we could use this information to predict future de novo mutations, because now we can see that we can tell which de novo mutations are more likely to occur. And there's actually more information that we found from this study that can also help us predict which de novo mutations are going to happen and where. So for example, back to this JBrowse screenshot, I didn't talk about this other variant that we had here right beside the de novo mutation. So this is a single nucleotide variant. It's coming from the father because you can see that the dam does not have this variant. And then you can see here that this variant occurs on the same read as the de novo mutation. And so this tells us that very likely this de novo mutation has occurred on the paternal haplotype. And so we were able to phase 15% of all de novo mutations that we found this way. And it's only 15 because we were working with short read sequencing. So our reads were about 300 base pair in length, and we needed to have an informative SNP on those reads to be able to phase the de novo mutation. So 80% of those phased de novo mutations are actually located on the paternal haplotype. And this is the same in humans as well. 80% of de novo mutations are occurring on this paternal haplotype that is being inherited by the proband. It was a little bit lower in dairy cattle, about 71%, but it's difficult to make this comparison because 15% of 704 de novo mutations is not a large sample size. So another thing that we looked at was the mutation rate and recombination rate. In humans, mutation rate increases near crossovers. And so just to remind you, during meiosis, when you have the paternal and maternal genomic information shuffling together, you have this double-stranded break of the DNA for this to happen. And probably what's happening is that repairs aren't completely made and finished before meiosis is completed. And then those mutations become permanent. And so we tried to look for the same in our study. So we looked in line 1, and we couldn't do exactly the same design as in humans. They had 5,000 trios where they looked at crossovers and de novo mutations. So instead, what we did is we used the offspring of the proband for line 1, all of them that we had that had been genotyped for 50K SNP array. Then we estimated the recombination rate in 1 megabase pair sliding windows across the genome. And this work was actually done by a master's student, Joseph. And what he found was that the average recombination rate in the surrounding 1 megabase pair of windows that surrounded the de novo mutations was over 15 times higher than the average recombination rate in the 1 megabase pair sliding windows across the genome. So we definitely see also not as in a fine scale as humans, but we also see, and this is the second species to identify this same phenomenon that's occurring between mutation rate and recombination rate. One other thing that we did to look at our de novo mutations and try to characterize them a bit more is that we used Ensemble, their variant effect predictor. And so we can break these de novo mutations up by their consequence. And then we also use this, it's called a PCAD score. So it's a CAD score, conservation score, that helps you prioritize and identify those variants that could have more potential to disrupt the biology of an individual. So you should compare the PCAT scores within each consequence, and the higher the PCAT score within the consequence, the more likely, or the more we should prioritize this particular variant. So you can see we found a number of missense mutations, splice donor stop gain, upstream and downstream variants that could have an effect on an individual, and they indicate with this high PCAT score as well. And I think that's really interesting to note that, you know, out of 41 trios, we do find a handful of these variants that could potentially have an effect on an individual. So I started to dive into the literature a bit, just out of curiosity to see some of these de novo mutations that were occurring in genes. And so I looked at the top 5. These are from top 5 PCAD scores from line 2 and then line 1 on the bottom here. So you can see that the variants are in genes that are involved in a number of different things. Could be related to body size and weight, stress response, immune response, and we also see morphology and lethality during fetal development. So interesting to see that, yeah, again, out of 41 trios, we are seeing de novo mutations that could potentially have an effect. So I also wanted to talk a bit about mosaic mutations as well, because when you sequence a trio of all somatic tissue, you could be potentially finding also these mosaic mutations, which are not de novo mutations. Well, they are new mutations, but they're not possible to be inherited by the next generation. So, as I mentioned before, de novo mutations occur in the zygote, or they occur in during gametogenesis, but our mosaic mutations occur a little further along in development, and then you can see that it ends up, you have an individual that has some cells that carry and some cells that don't carry this mutation. So when we look here at the reads, the frequency of reads supporting the de novo mutations in all of our de novo mutations found in trios where we use just somatic tissue, we would expect this supporting read frequency, the average, to be about 0.5, but instead we see it's a bit lower. And it's because we see that there's an increase in these de novo mutation candidates around this 0.2. And they found the same thing in the dairy cattle study as well. And it was actually, they were able to show that these were these were mosaic mutations that were causing this. And then just for your interest, I guess, we also had a few other trios where semen was sequenced for either the sire or for the proband, or for both of them or for one. And this changes which de novo mutations you can expect to find. It depends on the timing of when they've occurred. And I think it would be interesting if we had more trios of each of these types, because then we could better study the timing of de novo mutations. But unfortunately, in our case, we couldn't do that. We just didn't have a big enough sample size. So how can we tell between what is a mosaic mutation and what is a de novo mutation? We can do this by confirming that the de novo mutation has been passed on to the next generation, so the offspring of the proband. So we did this a few different ways. Initially, we wanted to estimate a false positive rate. So we took a sample, a random sample of 30 de novo mutations, and then we tried to have the lab to genotype these in 8 to 10 offspring of the proband. So you can see 17 de novo mutations were validated. This is quite a low number, but I have to say that we found, just anecdotally speaking, that our de novo mutations are often occurring in regions where there's a lot of repetitive elements in the sequence, and it's really difficult then to design primers and try and genotype these de novo mutations in this way. So our false positive estimate is not really, I don't think it's really something that can be relied upon here. And I think that it should be quite a bit lower than what we see here. But so we found that 17 de novo mutations were definitely de novo mutations. That's positive. So we also looked into some of the sequence data that we had as well. So line 1 had some related trios. So where a proband was a sire of another proband. So then we could look into that offspring and see if it had these de novo mutations had been passed along. And we found about 19 de novo mutations this way. And well, we were able to validate them. And I mean, we only had a couple or 3 trios that worked out like this. In line 2, in contrast, they had 10x sequence data from some key bores from the same line, from a different study that they had done. So we looked for these de novo mutations that we had found in line 2 to see if we could find them in any of the descendants of the probands from the study. And we found 63 de novo mutations that we could validate this way. And I just want to show you this really nice example from line 2. So here we have our trio from our study that was sequenced for 30x coverage. And yeah, you can see we found 17 de novo mutations in our proband. And when we looked at the sequencing of key bores, which are these squares in the darker color from the 10x coverage, you can see that we've identified 13 de novo mutations in the descendants, which I think is pretty good considering only one of these descendants were actually a direct offspring of the proband. And so I think the other really interesting thing that we can see here is that as the generations go on, so you see 1, 2, 3, 4, and probably by 5 and 6, all of these de novo mutations will be gone because they're really following the pattern that we would expect to see if no selection pressure is being put on these de novo mutations at all. So this is a really nice example, clear example of what is happening to these de novo mutations within this line. So to conclude, we were able to detect these single nucleotide variant de novo mutations in pigs using sequence data. And we were able to validate de novo mutations, and examples like the last one that I just showed you indicates that probably most of the de novo mutation candidates we found are true. We found an average of 19 de novo mutations per individual. This rate is probably a bit lower, but we were quite strict with our filtering because we want to make sure these de novo mutations are actually real. And I think we can see quite nicely that mutations are not random, and it would be really interesting to see if we can use this information in the future, information on DNA and other genomic features that could help us to maintain some genetic variation and improve the sustainability of breeding programs. So thanks for joining today, and I'm happy to take any questions or discuss any of my results. And please feel free to also reach out to me. My email address is here at the bottom here.
Great. Thank you, Chrissy. Does anyone have any questions? Not seeing any as of yet, but I will get things started. So yeah, Chrissy, you mentioned at the start that these de novo mutations were mainly seen in binary traits to do with health and fertility. Is that correct?
Yes.
Yeah, so were all of those detrimental effects, as in animals died, or yeah, infertility in males, as you said, they were always negative, or were any of them actually seen as being positive effects?
Yeah, so in humans so far, they've only studied those de novo mutations that have negative effects. So they're only looking at these traits that phenotypes that are probably not wanted. So yeah, that's And I think theoretically, we would expect that more of these de novo mutations would have negative effects, but it is possible that there could be some having positive effects.
And then to lead on to that is, as you said there, you feel like most of these effects are going to be negative. Is that a big like crux in the argument for doing this kind of work or trying to study it, that people might be turned off it because majority of the effects are negative? So if you want to breed these de novo mutations into the population that, as you said, a lot of them are negative. So is that something in the industry that they're afraid of kind of going down this route because of that, or not so much?
So I wouldn't say that people are afraid to go down this route. I think the real focus should be that that we're losing these de novo mutations at a faster rate than we have ever before. And we have relied on this variation to keep improving animals this whole time. And that, at a certain point, it might be that we run out. And so the question is so how do we maintain these variants, but at the same time try and not keep any of those negative effects and try and enhance and try and encourage these positive de novo mutations in our populations.
Okay, thanks. I think there is one question here in the chat. I'll read it out from Elizabeth Bolding. Chrissy, what benefits would a company obtain from 10x sequencing of key individuals in their breeding stock? Is 30x necessary? Most companies are still using SNP chips.
Yeah, so I think in the case of line 2 here, where they did have this 10x sequencing of key individuals, I think this was part of their own research that they were working on. I'm not sure exactly what they were doing. But one thing that I can definitely note is that in the 30x, I really think 30x is needed to find these de novo mutations. You can't really go much lower than that because it becomes very difficult to confirm that they're real. And I did struggle in finding some of these de novo mutations in the 10x data because it's just not deep enough. And so when I was looking and following up on these de novo mutations, I did actually have to look at some of raw sequence reads and just say, okay, yeah, the genotyper didn't find this de novo mutation, but you can see that it could possibly be there. So 10x is not adequate for this genotyping these de novo mutations.
Okay, perfect. Thank you. Louise, do you have a question?
Yes. Hello, Chris. Very nice presentation and good to see you again.
Yeah, nice to see you.
Yes, I have I'm just curious on hearing a little bit more some of your thoughts about recombination. So, of course, most of the time we do want to increase the genetic diversity in our populations, but especially as we are working with livestock, like in commercial populations, most of the time we are actually relying on linkage phase between the QTLs and the markers. Yep. And I have seen studies where they have looked at the heritability of recombination rate, and in some cases it is actually heritable. So I'm just curious to hear your thoughts on how we might use that information in livestock breeding programs. Are we going to be selecting for increasing recombination? Should we focus on specific regions of the genome? What are your thoughts about that?
Yeah, so I've also seen these studies as well. And I'm a bit, this is another one of those fine lines, because I think it's dangerous probably to increase the recombination rate too much, because we could then get a lot of these negative de novo mutations, or we can even be getting combinations of the genome that we are not interested in having. But on the other hand, yeah, I think we also need to just keep it in mind as an option. And I think that we really need to study more these genomic features, not just recombination and mutation rate, but I think there's also some other associated features as well, GC content, repeats, all of this kind of stuff, and try and have a better understanding of what's happening in the genome before we start selecting on these traits.
Thank you.
Thank you, Louise. Do we have any other questions for Chrissy? Yes, Jerson.
Hey, Chrissy.
Hey.
Nice presentation. Thank you for that.
Thanks.
Yeah, I had a feeling and problem wrong in some of your slides and even during the discussion here that it might be a relationship between inbreeding and mutation rate. Does that make any sense or?
Yeah, so that's another question that we've asked as well. So my student, master student Joseph, he also looked at runs of homozygosity as well to see if there was a relationship. We didn't find anything, but again, we were only using SNP data, so it's kind of hard to get like really good resolution on that. But yeah, I think that it is possible, like this is possibly one other genomic feature that we should be looking into as well.
Yeah. Okay, thank you.
Thanks.
Okay, any other questions then? Let's see. Okay, I'm not seeing any, so I'd just like
I
to
see
take
one in the chat actually.
Oh, sorry. Thanks for spotting it. Yes, from Rick. Dr. Chrissy, thank you for your presentation. I would like to ask if there is any study about those behavioral effects of de novo mutations and if they have been reported in other species.
So to my knowledge, it's just been schizophrenia and autism in humans that have been studied, and they haven't looked at any other species or any other diseases or disorders either.
Perfect, thank you. And Chrissy, one more just even from me. Do you feel like for like livestock species, which what we're more so interested, it's going to start with more so looking at disease, health kind of traits before we could ever kind of move this towards like production traits or traits that are on a continuous scale?
So actually, I think that I know that we are already starting to look at complex traits. I have a master's student who's defending next week, and she has looked at structural variant de novo mutations, and she was able to look to see if there was any associations between any of these traits that we had. So we don't have any records on disease or anything, but we do have records on production traits and reproduction and functional traits as well.
Okay, great. Elizabeth has another comment in the chat. Chrissy, when is your seminar in IB? I was later.
Yeah, yeah, that's in March 17th.
Okay. Okay, perfect. So if there's no other questions, which I don't see, I'd just like to thank you again, Chrissy, for the great presentation, and we'll see everyone back here again next week. Thanks, guys.
Thanks.